thanks for the nice introduction. And you basically, that's basically what I'm going to talk about today. And thanks for the invitation. And it's a great pleasure to be here. So I've been on campus a few times before and working on some collaborations with the Biotechnology Center. At that time, Franco Serena was still here. And so we collaborated on some of the rework, bio rework, and that started actually our work on the building genes using the microarray derived oligonucleotides. So it's a great pleasure to come back, and actually this is the first time I visit the biochemistry department, and it'll be nice to make new friends. So today I'm going to talk about one of the technologies that we have developed, which is the called integrated high-throughput on-chip gene synthesis platform. And then we're going to talk about some applications of this technology in the building genes and genomes in a large scale. So as I mentioned, my group has been focusing on technology development for the field of synthetic biology. And as you may know, that synthetic biology is a relatively young field, and with many promising applications in a lot of significant areas, such as bioenergy, making chemicals, materials, agriculture, medicine, environment. And it's interesting to point out that to realize all these significant applications in these many different fields, you essentially need only to manipulate one molecule, which is DNA. And therefore, it's critical to be able to design and synthesize DNA molecules in order to realize all sorts of different kind of functions. So that's one of the driving force of why I did this, working on the technology development in this field. So as you know, DNA, depending on the size, can encode many different functions, right? From the smallest a single gene, right, in the length of 1 kb average, to all the way to full genomes, right? So beyond single genes, at 10 to the 4th level, you can have big plasmids, genetic circuits, right? And if you can synthesize DNAs that large, you can essentially build networks, pathways, and plasmids to do a lot of functional studies. And next level above, if you go to 10 to the 5th, you can have essentially small viral genomes, and even a minimal genome. You can actually pick functional genes and assemble them into a self-replicating genome, the so-called minimal genome, to prove that you can actually understand how life works, right? If you can build such a genome that can self-replicate and itself. So that's a proof of, you understand life. And if you go the next level, go to 10 to the 6th, you can essentially have the one of the smallest bacterial genome, and from that on, you can build other types of higher, bigger genomes. So, in order to study and engineer all these systems, a crucial technology is to design and also to fabricate, right, these sequences from scratch. And so people have been doing DNA synthesis from the beginning, right after the discovery of DNA double helix structure, right? And one of the first person that to start DNA synthesis is actually Dr. Krana, and he spent 10 years on this campus. And he won Nobel Prize for his work, partially on the DNA synthesis. So he's one of the first person who actually started this whole field of DNA synthesis. And so from that time on, and people start to build larger and larger constructs using a slightly different chemistry than what was initially done. And so until today, just a year before, actually people have been able to synthesize not artificial genome, but one of the smallest genome. Is a replica of one of the smallest genome, which is about have 1 million bases, right? And they put that back into a cell, which is have been extracted of its own genome, and prove that this synthetic genome actually can keep the cell alive and functioning. So that's one of the first proof that show that synthetic genome actually can have the necessary function to support life. So that's basically a brief history of DNA synthesis. And it's interesting to point out that all these years, the chemistry involved in DNA synthesis is basically the same, right? The phosphoramidite chemistry. And the synthesizers are developed based on that. But it's still, even though people can synthesize a genome as big as 1 million bases, it's still very tedious and very expensive. And only maybe 1 or 2 lab can afford to do that, right? So that's obviously not sufficient for the whole field of synthetic biology to develop all kinds of different functions. So to do that, one of the critical technology to develop is actually the large-scale, high-throughput automated gene synthesis technology. And so that's what I have been doing. And if you compare the DNA synthesis technology, the development of DNA synthesis technology with the sequencing technology, actually you can see that they are pretty similar. It's just that the DNA synthesis technology has been far lagged behind the sequencing technology. If you look at the development of sequencing technology in the 1990s, people are pretty much doing sequencing by hand, right? So you do the reactions, you run the big sequencing gel, expose it, and then read the lengths one by one to get the sequence. And in the 2000s, people developed the automated DNA sequencer. So you can sequence pretty much 1,000 or less than 1,000 bases in this automated fluorescent reaction. So the computer can read it, right? So after sequencing, you can get a report from the computer. And now we have single molecule or whole genome sequencer where you can sequence a bacterial genome in a few days, right? For a few $1,000s. So the technology for DNA sequencing has been evolving pretty fast. And so where is DNA synthesis technology right now? I think it's still at probably this stage. We have automated DNA synthesizers, but that can only synthesize oligonucleotides about maybe 100 bases long, right? And from that on, if you want to build genes, which has normally 1,000 bases, you pretty much, it's a manual process, right? You have to do it manually in order to assemble those oligos into genes. And so in that sense, we are still at around this stage, and we are far from building an automated synthesizer which you can use it to synthesize full genomes or large-scale production of many different genes, right? So we are still far away from that. But at least this, chart tells us where we are for DNA synthesis. So what are the major challenges in gene and genome synthesis? So in my opinion, there are at least three. One is the cost and the throughput for oligonucleotide synthesis. It's still very expensive even to make the short oligos, right? And the next is eliminating gene synthesis errors because the chemical reactions involved in DNA synthesis has efficiency issues, right? It's not 100% efficient, so every base you add, you accumulate errors. So how to get rid of those errors is very challenging. Right now, people are using cloning and sequencing to get rid of, to pick the right sequence, but that's very costly and time-consuming, right? So that's one bottleneck. And if you talk about synthesizing whole genome, which is normally above 1 million bases, how to assemble and handle those long sequences is another challenge, right? Because it's, I mean, the longer you get, the fragile the sequence, the molecules will become. So it's very difficult to handle that, and also how you can put that back into a cell or into a system where you can jumpstart the whole process, the whole function of the genome is another challenge. So the synthesis of that first minimal genome proved that people can have accumulated some knowledge to handle this issue, but moving beyond that, if you want to synthesize E. coli genome, is still a challenge that people haven't been able to prove that it can do that. So my work, I mean, for this talk, I'm only going to focus on the first two issues and talking about some progress we made in those areas. So what's the problem of this, the conventional gene synthesis technology that are currently used? So if you look at the process for the conventional gene synthesis, the first step is oligosynthesis, right? We have automated DNA synthesizers that can make maybe 100 base long oligos, right? And that takes about half a day to a day. And then you need to purify the oligos and assemble them into genes, and that takes another couple of days. And then the big issue of error correction or picking the right sequence, right? And that is the tedious one. You have to clone your gene into a vector and put that into E. coli, for example, grow them up and sequence them. If you're lucky, you can pick the right clone right from this one round. If you're not lucky, you have to probably repeat this cycle, and that takes more time. And or you have to do a site-directed mutagenesis to correct those errors. So that takes even more time. So right now, I mean, the price for DNA synthesis has dropped significantly from initially it's about $28 per base. Right now it's less than $1, half dollar per base. But still, if you're talking about large-scale synthesis or full genome synthesis, it's still very costly, right? So so what we are trying to do is actually to see if we can simplify this process or automate this process. For example, integrate the whole process of oligosynthesis and the gene assembly into one step, right? And then try to increase the accuracy of gene synthesis and eliminate the error correction step. So this will drive the cost down and also significantly reduce the time for gene synthesis. So that's the goal for our research. So let's talk about first how to integrate this whole process. So at that time, we thought about this question, and so one of the technologies that we can borrow actually was the microarray technology, where you can synthesize thousands or even more different oligos from a single chip, right? But the issue is each oligo, I mean, even though you can synthesize many different sequences, but the amount of each oligo is still pretty small and not enough to drive the gene assembly reaction. So we have to come up with ways to, somehow amplify the sequences and also figure out how to assemble genes from this big mixture of oligos. So at that time, this is one collaboration we did with a group in Texas, and using the microfluidic DNA array to synthesize different oligos in different chambers. And this is controlled by the digital micromirror array, which I think Franco Serena was one of the pioneers for this technology. And so at that time, I also collaborated with him. So we published around the same time on this technology using the DNA microarray to assemble genes. So this is the type of microarray we used at that time, using the so if you look at the array, is pretty small, right? The size of a fingernail, but it can synthesize thousands of oligos on a single chip. And then to amplify the amount of oligos, we developed this, having a common primers on both ends, and then you can using a PCR-like reaction to amplify the oligos, and then you can remove the primers at the end with a type 2S restriction enzyme, and then you release the clean oligos. You can use that for to build genes, right, using those oligos. So in that way, you can get cheap oligos from, inexpensive oligos from the chip, and it proved that we can use this method to synthesize multiple genes at a time. And but after that, we realized that this technology still has limitations, because you have, although you can synthesize many different oligos from one chip, but the pool is probably very complex, and it's not easy to, efficiently utilize every sequence to build many different genes as you want, because of the complexity of the sequences. Since then, people have been developing different ways to get around that. And I think there are companies right now, using this technology to and the different approaches to efficiently utilize different areas of the chip to get as many as genes out of the chip. So some strategies, you can use bioinformatic tools to design different primers, right? Different PCR primers, so you can selectively amplify different regions of the chip into different pools of oligos, so you can assemble them into different constructs. So that's one strategy. And there are other strategies that companies using to build tags into these oligos and then try to assemble them into different sequences. So we took a different approach. Actually, we divided, the strategy we used was actually to try to divide the chip physically into different areas, or different wells or chambers, if you will. So in each chamber, we can have a smaller group of oligos synthesized in each chamber, right? And then we can amplify the oligos and then use the free oligos to assemble them into genes directly, and not taking out into a tube, but directly on the chip, right? So in that case, we not only simplified the reduced the complexity, but also integrated the whole process of oligo synthesis with gene assembly on the same chip, right? That's the original goal we tried to achieve. So in this one strategy, we hope we can actually realize that goal. So for this design, we used different technology than the original, the micromirror array type of chip. So this time we used inkjet printing system technology. So just like the printer you have in your home, right? Instead of printing ink, we can put different monomers, phosphoramidize into the well and then print them on the chip to make different oligos, right? The chemistry, another advantage of using this is the chemistry is the same as the phosphoramidite chemistry used in the standard automated DNA synthesis, so you don't have to have special chemicals to do that, right? And that simplified and also reduced the cost of this process. And we proved that we can do this pretty well. And also, in order to achieve this in situ synthesis and assembly, we have to make our own chip. So the conventional type of chip is, use glass or silicon. But here, in order to make it easy to fabricate, we used a plastic chip, a COC chip, so you can easily mold what kind of features you want on the chip. And then the common microarray technology for DNA synthesis has an issue of higher error rate than the conventional DNA synthesis because of the surface issue. If you spot something on the surface, the next step you put on may not hit the same area, right? So the boundary shifts between each step, and that creates a much higher error rate than the conventional column-based synthesis. So to correct that issue, we actually deposited silicon dioxide spot on the plastic surface. So only on the spot you can grow oligos, right, on that. And in that case, you keep the boundary clean and reduce the error rate. So in this way, we can synthesize oligos which has a similar error rate compared to the conventional automated DNA synthesizers, which is about 1 in 5 or 600 bases. So that's pretty good. So with these modifications, we were able to put all the reactions on the same chip, but still we have to develop a new kind of reaction in order to streamline the oligosynthesis, amplification, and assembly, right? And have them in a single well. So to do that, this is the strategy we used. So we synthesized different oligos in the same well, and then at the end of the oligo, we have a common primer. And using this primer, we nail a primer onto it. We can extend, right? We can extend and make it into double-stranded. And also at this site, there is a nicking site. There is a nicking site here where a nicking enzyme can come in, recognize the site, and make a cleavage, a single-strand cleavage here. So once the nick is made, another polymerase can come in and start extending from this nick, right? And also this polymerase has the DNA, the strand displacement activity, so it can displace the strand in front of it and release it into the solution, and that strand can participate into the DNA, the gene assembly process. So in this single well, you have the mixture of enzymes and optimized buffer. You only need to change temperature, right? From this is a thermal isothermal amplification, and this is a thermocycling amplification. So only by switching the temperature mode, you can switch from amplification to assembly, right? So in a single well, you can actually assemble genes out of it, right, without changing buffers. So that essentially integrated and automated the whole process of gene oligosynthesis and gene assembly. So we pretty much achieved the first goal, right, of integrating and automating the process, also miniaturizing the process. So the second goal is actually to increase the accuracy of synthesis to essentially eliminate the error correction, the cloning and sequencing process. So to do that, we developed another reaction which take advantage of one enzyme which is isolated from celery plant. And that enzyme has the activity of recognizing the mismatch structures in DNA, right? If you have a mismatch, either it's caused by mutation or by deletion or insertion, this enzyme can recognize all those structures, the mismatch structures, and cleave it, right? If you have a high concentration of enzyme, it can actually make a double-stranded cleavage and after the mismatch site. So that's a very useful activity. So we took advantage of that activity and integrated into this gene synthesis process. So we just added one cycle of this error correction reaction into it, and that can essentially eliminate most of the errors in it. So let me run this through with you. So this is the normal gene assembly process, right? You start from oligos, assemble oligos, PCR extension into full-length genes, right? And at this step, you still have a lot of errors in the full-length genes. And to remove that, we added this enzyme. We did a denaturation and re-anneal those so the correct sequence can anneal with the incorrect ones and expose the errors, right, in the sequences. Either it's insertion, deletion, or mutation, right? They all form this mismatch structures, which can recognize by this nucleus. The commercial name is called severe nucleus, which can recognize the mismatch and make cuts right after it. So it exposes the mismatch sequences. And then there's a 5' to 3' exonuclease activity in the reaction, which can remove all these mismatch bases, essentially remove all the errors from it, right? And then you use the error-free fragments and reassemble them into full-length genes, right? So through this one cycle, you pretty much eliminated most of the errors. And so we sequenced a lot of those sequences before and after error correction, and we show that through one cycle you can pretty much reduce the error frequency from 1.9 per kb to 1.9 per kb. It's a 10-fold reduction. And through further optimization, and if you can also do multiple runs of this reaction, we can pretty much reduce the error rate to below 1 in 8,000 bases. And that's pretty much good enough for a common gene synthesis, right? So meaning that if you compare the two error rates, so if it's 1.9 per kb, essentially if you want to build a sequence longer than 1 kb, you pretty much don't have much any sequence left in the population which are 100% correct. And if you have an error rate which is 10 times better, you can still, if you even build a 10 kb sequence, you still have more than 30% correct. So that's the difference, how big a difference you can make with this difference in error rate, right? And if you want a visual demonstration, here is a GFP gene that we try to synthesize with or without this error correction. So without error correction, I mean, only half of them are fluorescent. With error correction, more than 90% are fluorescent. So that's the difference that error correction can make in this process. So hopefully, I mean, we are still trying to improve this process. I mean, this enzyme is good. It can recognize all types of mismatches, but it still has an issue of efficiency towards different types of errors, as you can see from this study, right? It recognizes deletions, insertions better than mutations. So if we can improve that, make it more sensitive to mutations, we can further improve this process. So that's an ongoing study we're doing. Hopefully we can use this type of approach to eliminate most of the errors. Of course, there are other approaches, such as my former advisor, George Church. I mean, he's trying to use high throughput sequencing to sequence every article and pick the correct one and then assemble them into genes. That's another approach, if you can afford to have a genome sequencer in your lab. So one of the goals for this research is actually to build a machine that you can, put in your gene design and push a button and you get a gene product. So that's just like a PCR machine. On your benchtop, you can have your gene design and synthesized for you. So that's pretty much the technology development that I'm going to talk about. If I still have time, I'm going to talk about, there are many, many applications you can do, right, with this type of synthesis capability. You can make, for example, large libraries of genes, or you can make whole genome, right, genome synthesis. And so today I'm going to just talk about one application we did, which is on the regulation of protein expression, and focusing on the codon optimization part of it. There are different ways that you can regulate expression of a gene, right? Some of the common ones include promoter, right? You can use different promoter sequences to change its strength. Also, you can modify the ribosomal binding site, change the ribosomal binding activity, affinity, so give you different strengths of initiation. And also, you can change codons of the gene, right? Using different codons, you can change. People have been shown you are able to change the protein expression level just by changing the codons. However, the rule for codon usage is not quite clear. There are softwares out there, or companies use software to design the codons for you, but half of the time, it didn't work. If you try this, send to the synthesis companies, okay, you optimize this for me, and they will send you back a sequence, and you try it yourself. So half of the time, it didn't work. It shows that people just don't understand completely the rules involved in codon optimization. And there are many factors involved, which I'm not going to go through every one of them. But GC content, secondary structure, or the adaptation, codon adaptation indexes are all shown to be key factors in that. But just how they play together is a big issue. How you design the software that can incorporate all these factors into it is a big challenge. And people have been shown that in the gene sequence or RNA sequence, if you just change one codon, and that can totally screw up the whole translation process. And how can you design a software to predict that? So it's very challenging. So with our technology, we were thinking, okay, probably we can use this high throughput synthesis approach to solve this by design and synthesize many different versions of it and see which one works best, right? So if you accumulate enough data, you can probably derive some kind of rules out of it. So to do that, the first step, I mean, you have been able to, you need to be able to synthesize big libraries, right? Codon variants libraries, and in order to clone that into a vector, I mean, there are some strategies that you can do that, but we developed our own because it's simple. So it's basically like a PCR. I mean, you have your vector and you have your library, right? And the only requirement is you need to have overlapping regions between the two, and then you nail them and extend them and transform directly into E. coli, and that's it. It's pretty simple, and it's not sequence dependent. You don't have to do digestion ligation because it's a complex library, right? It must have every restriction size in it, so you cannot use those kind of approach. But this is pretty simple. You just one round of PCR, you can pretty much get your library cloned. And also you can use this to assemble a number of fragments in one step into a plasmid and clone it into and grow that into the cell. And we proved that it can work pretty well to close to 10 kb, the whole construct. So using this approach, we did a pilot experiment to see, okay, whether we can, just by varying the codon usage, we can achieve a spectrum of different expression levels. And so we used the LAGZ alpha as a demonstration, and it showed that it worked pretty well. I mean, this is the wild type construct, LAGZ alpha. You have very even, uniform distribution of colors. And this is the codon variants we synthesized and cloned into them and played them out. You can see there are great variations of colors, right? Meaning the different expression levels of this single protein. So that's a demonstration that, I mean, this approach works for altering the protein expression level. So here, this figure shows is a demonstration that we designed over 1,300 different codon variants, and we measured their expression based on the color, the LAGZ color. And so here you can see, I mean, we can have very strong expression all the way to a very low expression level and everything in between, right? So it's a demonstration that by altering the codon usage, you can pretty much get every single level of different expression, protein expression level. And this is a distribution of all these constructs, and you can see the expression level. It's very interesting that this is the wild type expression level, and most of the sequences, I mean, have this expression level, and about a third of them have expression level higher, and 2/3 are lower. So we are in the process of trying to figure out what factors are involved in this process, and also we are doing a number of different proteins using this approach to see if we can find a common rule involved in this process. And also we're doing this in different organisms, different cell types, to see if there are, I mean, there must be variations, but we just want to see what are the variations, what are the causes of these variations. And here is just a video showing you the panel I showed you before, the expression level and speed of these different constructs. So this is a demonstration that, I mean, this approach can generate all types of different expression levels for the same protein. And to make it, I mean, practical to work on a real problem, we happen to have a collaboration with Kevin White at the University of Chicago Genome Center. So they are doing the ENCODE project, where they need to make antibodies for all the Drosophila transcription factors, but after they tried that in E. coli, they found at least half of them don't express in E. coli. So he asked me if I can help using this technology to express all those sequences that normally do not express in E. coli. So I said, okay, I can try. So he gave me 75 of those. And so I went through the whole process that I just described. So you design the library, you make them, you synthesize libraries. So each band is a library for all the codon variants for a single gene. And so we made all 75 of them. And so we made the whole library and screened them and picked the best expressing ones. So as you can see, compared to the wild type, which pretty much don't express at all, after optimization, you can have a fat band there, right, which is about more than 50% of the whole cell protein is that band. So we demonstrate that we can make all 75 work using this type of approach. And so it would be nicer if after we do many different genes, we can figure out the rules and then design a software so you don't have to go through this process. You can just design one and get the highest yield you want. And also this technology is also good for picking the intermediate levels. This have some usage in synthetic biology where people build synthetic networks where they want to regulate the expression of each component in it, right? So normally in that you have a promoter that drive a string of proteins, but how can you manipulate the relative expression of each protein? Using the same promoter, right? This is probably one approach that you can use. And for that, you probably don't want the highest expression for every one of them, right? You want intermediate levels. And I think this approach can probably provide that kind of solution to that problem. So with that, I want to that's pretty much all I want to talk about today. And I would like to thank people who did the work and also the funding agencies. So after I talked about this work, a lot of people just want to try it out and want to take advantage of some of this technology. So obviously this cannot be done in an academic lab, right? I don't have students that can constantly work on other projects. So I set up a operation that if you have this kind of need, that I can probably offer some help on that. So with that, I'll stop here.
How many independent gene assemblies can you do in one of those chips?
How many assemblies I can do on a single chip? All right. So currently we have designs that you can have 30 different constructs on a single chip, but you can very easily enlarge that or make it even more, right? Mm-hmm. To structures of proteins? So you're talking about the final three-dimensional structure of a protein and not the RNA secondary structure? That's a very interesting question. I don't think people have been able to address that issue with the current synthesis technology because it's pretty hard. You need to have a significant number in order to do this correlation. But in one of the slides I showed you, so in this one, it's an accidental discovery where there's only one codon change, and that can alter actually the final protein structure. Not necessarily. I mean, but later people figured out it's probably caused by the folding process, right? So if you have one change of codon, one codon change, and that alters the speed, the ribosome migrate along the mRNA, and that causes a change in the protein folding process and makes the protein less stable than the original one. But you're right. I mean, that's probably one of the initial proof that codon usage can actually affect protein structure. But how, people haven't been able to dig into that. Yeah.
So, you stated that the gene synthesis is now 30 cents to $1 a base. Is that
Yeah, depending on the length. If it's longer ones, it's more expensive. Yeah.
Is most of that cost the oligosynthesis?
No, oligosynthesis is about well, actually, 10 cents per base if you buy oligos. But it's it's probably less than a third of it is oligosynthesis.
10 cents a base, did you say?
Yeah, less than a
So
third
about
of
a
it.
third of the third of the fifth of the cost is really the oligo. So
Right.
is there much error correction going on in the when you buy oligos off a machine instead of off a chip?
No, there's not much error correction going on. Most of the synthesis companies, they just cloning and sequencing, that's it. Yeah, they don't do much error correction.
What's the next big step?
In synthetic biology or in just
in your field?
In my field? Well, actually, if you can, suppose you can do large-scale chip, inexpensive DNA synthesis or gene assembly. Suppose you can, synthesize anything you want. I think the next step is, one is to find out the rules, how those sequences can control the functions of life, right? Figure out that, and also applications. You can, once you learn the rules, you can use the rules to help you build different, for example, design different proteins, enzymes, or design different genomes. You can achieve different functions with those designs. I think that's that could be one of the directions. Actually, a lot of people are working on that, but there are probably further, more significant developments in the future. Yeah, probably only people like you can predict. So.
Between creating life and making genes, where is the limit of what size DNA you can physically assemble?
Limit.
How are people addressing that?
Right. I guess, for synthetic DNA, it's the length could be a bigger issue than the in vivo DNA, because in vivo you have proteins, factors bind DNA to protect it, right? And in vitro, it's just a bare DNA you want to synthesize, and the longer it gets, the fragile it gets, right? The more fragile it gets. So it's the handling becomes the issue, right? And also assembly becomes the issue. So right now, the assembly for bigger chunks is mostly done in vivo. You put them into yeast using the yeast recombination system, assemble them. In vitro, there are enzymatic reactions that you can do to assemble them, but the length, there is a limit, all right? And people can do over 100 kb using an in vitro system, but most of the larger ones is done in vivo, in yeast. So, so how big a construct the yeast can handle is probably, remains to be determined. So I guess there might be some limitations. Yeah, we tried. We actually varied a lot of the factors in here. Try to build libraries based on the variation of a single factor, like, for example, GC content, right? We can try the same gene, we can do 20% GC content versus 80% GC content and see how that can affect expression levels. And there is a big influence, just the GC content. Mm-hmm. Yeah. There's also a limit on that. If you use a high GC level, it gets very difficult to assemble or synthesize the sequence. Using the chip technology, you can get down to lower than 1 cent per base, or even lower, for the optimization. Right.
There are no more questions. Let's thank Dr. Tia.
Mm-hmm. Thank you.